Hello everybody and welcome to lecture 8. This is actually lecture 1 of the 3 lectures I'm going to give on cardiovascular physiology. Okay, so we're going to start by talking about some of the basics of circulation here in terms of the circuits. So we have pulmonary and systemic circuits. What this means is that this is the blood that's moving through the lungs versus the blood that's moving through every other part of the body. So we call that the systemic flow. And so what you're seeing here in the cartoon is the part labeled in blue there is actually the venous blood, and that's oxygen poor blood. All right, oxygen poor. It's not devoid of oxygen. It still has oxygen. It just has less oxygen because this is the blood that's returning from the tissue to go back to the heart, and the tissue has already taken the oxygen it's needed or that's required to meet its metabolism. In the meantime, it's also picked up CO2, which is one of the byproducts of the tissue metabolism, and so it's got to return that blood to the lungs. So it does so by going to the right side of the heart, which is the right atrium, via the superior vena cava, which is really coming from the upper extremities in the head, and the inferior vena cava, which is coming from the lower extremities and the abdominal region. And then you see from the right atrium communicates with the right ventricle, so it distributes the blood to the right ventricle to the lungs. And so here we have our pulmonary circulation. A couple of definitional terms. What we have is anytime we refer to something as an artery, that's blood leaving the heart. A vein is any blood returning to the heart. So you'll notice on the right side of the heart, there's blood leaving the right ventricle to go to the lungs. So that is actually a pulmonary artery. And then the pulmonary artery will, of course, divide into capillaries that go throughout the lungs in order to exchange gases, get rid of the CO2, pick up the oxygen, and become oxygenated and return that blood to the left side of the heart, to the left atrium, and then ultimately to the left ventricle, which now is oxygenated blood. So we call this, under normal circumstances, fully oxygenated blood. And then from the left ventricle, that's going to pump it out into the systemic circuit. The systemic circuit consists of the first branch, or the first artery, which is the aorta. And then that's going to branch many, many times into smaller arteries, into arterioles, and then into capillaries. Distribute, of course, the oxygen to the tissues via the capillaries. And then the venules will coalesce into the veins, into the superior vena cava, and return that oxygen poor blood back to the heart. To pick up more oxygen. So everything down here, which you just talked about, all of this is actually all part of the systemic circulation. Okay, so here, just to kind of impress on you the blood vessel volumes, I've actually shown you this chart, or something similar to this in lecture 2 when we were talking about capillaries, where I was showing you the cross-sectional area. You can see the cross-sectional area here in centimeters squared. And it's taking us through, the branches. So in the systemic circulation, we have the aorta, then the small arteries, arterioles, capillaries, venules, small veins, and then the vena cava. Now, you'll notice that, the aorta being a singular artery is, the largest artery in our body, but it's only singular. What happens is as they branch, their cross-sectional area becomes summed as they run parallel to each other. And so by far and away, the largest cross-sectional area is in the capillaries. Now, in that lecture, I talked about how the large cross-sectional area actually reduces velocity of blood flow going through that area. What I really want to kind of impress on you in this particular chart is just comparing the arterial side of the systemic circulation here, right? The aorta, small arteries, and arterioles. You'll notice their cross-sectional areas, and if you compare them to the venous side with the venules, the small veins, you'll notice the cross-sectional areas of the veins are much larger, and in fact, the venous side of the circulation at any given time contains significantly more blood than the arterial side. So about those veins, what we have is the veins have a greater capacity for blood containment than the arteries. So that's partly due because of the fact that they just have larger cross-sectional areas. They could accommodate a lot more blood, but they're also more distensible. So they can contain more blood to larger volumes. They're more distensible than arteries are. And the fact that they're more distensible, meaning their ability to passively stretch with pressure, means that they actually have a greater compliance. Okay, they have a greater compliance, so they could hold more volume than the arteries can. The arteries are a bit stiffer in structure. Okay, they have thinner walls, all right, compared to arteries. They're typically what we call more flaccid, less muscular, and they have less elastic tissue overall. Even though they contain all the same layers that the arteries contain, all right, which is an endothelial layer, subendothelial layer, smooth muscle layer, an externa, and all that. The layers are much thinner. They have far less smooth muscle, all right, and they can collapse when empty, and they also expand easily because they have really great compliance. And this is something, if any of you guys have ever tried to draw blood on a patient, some are easier than others, but in particular, dehydrated patients are quite difficult to draw blood from sometimes, especially in the superficial veins just underneath the skin there, because they can collapse. There's not as much volume there, and so they can kind of collapse themselves. Arteries tend to hold their shape. Okay, they tend to stay, in a more of a more rigid structure. So veins tend to have what we call a steady blood flow moving through them, as opposed to an artery, which is more pulsatile. And this would make sense because when the heart's beating and let's say the left ventricle is pumping, it's pumping blood into the arterial system. Now the arterial system, that means it's basically generating a lot of pressure from that pumping of the left ventricle. Every time it squeezes, that pressure is translated directly into the aorta, which also subsequently generates pressure downstream into the arteries and so on. But by the time you get to the veins, all right, the pressure is significantly reduced. And oftentimes what you have is the arteries will actually be pulsatile because of the beating of the heart. Every time the heart contracts, you're getting a surge of blood into the arterial system, and that's causing this pulsations that we can actually feel. So we know how to check our pulses, like radial pulses, okay, and so on. So we can feel that, we can palpate that. However, venous blood doesn't have pulsations. In fact, it runs very smoothly. And you can actually see this clinically if, let's say, somebody's undergoing surgery. If you were to nick a blood vessel and you weren't certain if it was, say, an artery or a vein, you could see by the way the blood is actually leaving the body or leaving the vasculature where it was, because arteries will be more pulsatile in nature, and venous blood tends to kind of ooze. Again, veins tend to merge to form larger veins, so again, they start as venules coming from the capillaries. And overall, they have a much lower pressure, again, because they're not connected, they're further away from the pumping of the heart of the left ventricle there. And if you look at the pie chart here, you'll see over here, the veins. This is showing you the distribution of blood through our circulatory system at rest, all right? And it's showing you, again, this is the systemic circuit, which is this part of the pie here, systemic circuit. And you can see the pulmonary circuit over here. And then the heart's in its sort of own category. And in the veins, 54%. So you can have anywhere from 54 even to 60, or even a little over 60% of your blood at rest could be found in the venous system. So it's a large volume found within the veins. And so the veins can actually act as a reservoir. Because they're so compliant and they can distend with all this blood, it can act as a reservoir for blood. So let's say we're to lose a little bit of volume or lose a little blood, the veins can actually constrict a little bit. They have muscle. They can constrict a little bit and increase the amount of blood that circulates through your heart and through your circulatory system to help keep the blood flow steady. Now, over here, you see the capillaries only take about 5%. The arteries only take about 11% comparatively to, say, the veins. The pulmonary circulation about 18% in total, and then the heart about 12%. All right. So really, at any given moment, you can see here from this pie chart that you're looking at about 70 or even greater than 70% of your blood is found within the systemic circulation relative to your pulmonary circulation. Okay, so now we have to talk about the position, size, and shape of the heart. This is actually very important clinically because it can change in size and position and so on, and this helps us diagnostically. So in terms of its position, the heart's located in the thorax, which kind of sits on the diaphragm, and you can see the diaphragm right about here. It sits on this muscle, the diaphragm, which we use to breathe. And it's situated between the lungs, the left and the right lungs. So this is the left lung over here. Here's the right lung. and it's in a part of the thorax we call the mediastinum. Okay. In this cartoon, you can see they've actually cut a little window here in some connective tissue so we can see the heart a little more clearly. And that tissue that surrounds the heart and sort of encompasses it is called the pericardium, which I'll talk about in some slides to come up. Now that pericardium, they cut part of it away so we can see some of the heart there. You'll notice the pericardium is also tethered to the diaphragm there. All right. The position of the heart itself, you'll notice that the tapered end of it down here points towards the left lung and actually into the left lung a little bit, and that's referred to as the apex of the lung. The base of the lung is more superior, and it's actually the superior portion of the heart that actually contains all the large vessels, where all the large vessels are connected to the heart. So the veins and the arteries and all that are on the superior portion of the heart, which is referred to as the base, which I understand can be a little bit confusing. All right. Again, here's the heart location, and what we can see here, this is actually in a cadaver. You can see some of the muscle right here of the diaphragm. This would probably be the liver just underneath the diaphragm. You can see the right lung over here. So the right lung, you can actually see some of the lobes of the lung there. You see the lobes. Over here, all right, so this is the right lung, this is the left lung. So in the mediastinum, which is this whole structure right here, we can see the heart. You see the apex of the heart down here. You see the base of the heart right here where all the major vessels are attached to it. In fact, here's the aorta. All right, you can see the pulmonary trunk right there. So pulmonary trunk. This is the aorta. All right, you can actually see here this right here is the superior vena cava. A little difficult to make out the inferior vena cava in this particular picture here. All right, and one other thing I want to kind of note on this is that you'll notice that the heart has this yellowish look to it, and in fact, a lot of there can be some adipose tissue on the surface of the heart there, and in fact, that's where a lot of what we call the coronary blood vessels, which are the blood vessels that supply the heart with blood, they can run through the surface of the heart there and through some of that adipose tissue. And in fact, what you're seeing is the sort of anterior aspects of the ventricle right here. This anterior part that faces like just underneath your sternum, that's primarily your right ventricle, is what we're seeing here, is the right ventricle. The left ventricle would be more lateral over here. So over here, this is the left ventricle. We only usually see that more towards the lateral side. The rest of the left ventricle is actually posterior, and we can't see it in the cadaver at this time. So anatomically speaking, normal physiological position of the heart, apex towards the left, base is more superior. This is substernal, or just behind the sternum. And the right ventricle faces most anteriorly, while the left ventricle is more posterior. So the pericardium, I mentioned the pericardium, which is the connective tissue surrounding the heart, a couple slides ago. And this is actually connective tissue that has two layers. So there's a visceral and a parietal pericardium. And the pericardium, peri just means around. So this is a connective tissue that surrounds the outside of the heart. And it contains what we call the visceral and the parietal pericardium. The visceral pericardium is adhered directly to the outer wall of the heart itself. And in fact, the visceral pericardium is also known as the epicardium. So the heart itself has three layers, and the epicardium is the outermost layer of the heart. And the epicardium is actually synonymous with visceral pericardium. They're one and the same. Okay. This is a serous membrane, so it's going to be secreting like a thin fluid, all right, that covers the outermost layer of the heart. The parietal pericardium is continuous with the visceral. So if you take a look at the cartoon here, particularly at the top corner here, you'll notice that the part that's connected directly to the heart here, it turns on itself and then becomes the parietal pericardium. And the parietal pericardium is not directly adhered to the outer surface of the heart. And so an interesting way to think of this is that in this cartoon here is if the fist is the heart, for example, take the fist and you took like a partially inflated balloon and you push your fist into the balloon. What's going to happen is The part that comes in contact with your fist is the visceral pericardium. That's the part of that balloon. But then there's going to be an outer part of the balloon that's not touching your fist, and that's the parietal. And then in between, you're going to end up with a little layer in between, or a little space in between. In the balloon, that's just air. But in the body with the pericardium, that's a very thin space that will be filled with a tiny bit of serous fluid, or like thin watery type fluid. And we refer to this as the pericardial cavity. That's the space between the visceral and the parietal pericardium. Now this pericardium also adheres the heart to the diaphragm, which you can see down here. All right. Now, some of the point of this is actually one to help keep the heart in the position within the mediastinum. And there's a little bit of fluid between the layers, so the heart's constantly working. All right. And so because it's constantly working and generating all this energy, it's also generating heat and whatnot, it can create some friction. And so this helps to actually reduce the friction within the thorax there by having a little bit of thin fluid secreted into that space and helping to maintain its position. And as you will see later on in cardiology, that tiny amount of fluid that's in that pericardial cavity could be greatly increased. You could have hemorrhaging in there and have bleeding in there, or large volumes accumulate in there and compress the heart. Something we call a tamponade, which I'll talk about in cardiology more. All right. Now, this is interesting because the cardiac muscle, this is doing a lot of work to contract, and ultimately the goal is to move blood through the heart and into the pulmonary circuit or into the systemic circuit. And the arrangement of that muscles, how the cardiac muscles kind of connect to each other, and the overall arrangement that they take in the heart is actually more of what we call a spiral orientation, which you can see here. If you take a look, you see it kind of looks like a spiral. And the reason that's interesting for that orientation is we have to think in terms of how it contracts and moves the blood out. So when the blood fills up these ventricles, right, and the ventricles, if we're taking a look at the heart over here, it's this part of the heart. These are the ventricles. Up here, all right, is going to be the base of the heart, which is the major blood vessels connected to the atria. But the ventricles are the ones that have to do a lot of the heavy lifting in terms of moving blood out of the heart and into the different circuits. And so what the heart does is it actually contracts from the apex. Remember, the apex is down here. It contracts from the apex upwards, because remember, all the major vessels are located at the base, which is superior. So it contracts downward and in a spiral fashion, so it squeezes and sort of twists like wringing a towel. It kind of twists and squeezes from the apex up towards the base. The purpose of that is so that anytime the ventricles fill with blood, we squeeze upwards towards the outlets, towards the arteries, so we can get them into the circuits, the circulation. And otherwise, if we were to squeeze everywhere at once, we might not get the most effective or efficient way of ejecting blood out of the heart. All right, and we especially don't want to do it the opposite way, which is from the base down towards the apex, because there's no opening or outlet from the apex. So, as I mentioned the chambers, we got to take a look at those. So if I was to make a section through the heart, all right, it has overall 4 chambers. All right, and we tend to look at the hearts in terms of right side versus left side and so on. So on the right side of the hearts, I'll show you over here. This is the right atrium. Okay, and the right atrium is connected to the vena cava. All right, you can see the vena cava over here. There's the inferior vena cava and the superior vena cava. And so this is the venous blood that's draining into the right side of the heart, into the right atrium. Then separating the right atrium right here is what we call the tricuspid valve, or otherwise known as the right AV valve. AV just stands for atrioventricular valve, meaning it's a valve that separates the atria from the ventricles. All right. And then next you have the right ventricle, which is this whole chamber down here. All right, that's the right ventricle. And then the right ventricle, its job is to contract and eject blood into the pulmonary trunk, which you can see over here. Pulmonary trunk, which leads to the pulmonary arteries and into the lungs. And once it gets oxygenated in the lungs, that blood returns then via the left and right pulmonary veins. Now this blood is now oxygenated because it just passed through the lungs, and it's going to move that into the left atrium over here, which we can see part of. All right, so that oxygenated blood coming from the lungs goes into the left atrium, Separating the left atrium from the left ventricle, which is right here, all right, is our left AV valve, otherwise known as our bicuspid, or more commonly, probably the mitral valve. And I'll get back to that. All right, so that's the mitral valve. The left ventricle's job is to actually eject that blood, that oxygenated blood, into the systemic circulation. So it's going to be pumping into the aorta. And so you can see the aorta right here. All right. And so if you take a look closely, you'll notice the muscular wall of the left ventricle is much thicker than that of the right ventricle. And that's because the left ventricle's job is much harder than the right ventricle's, because it has to overcome a lot more pressure, because it has to pump blood through the entire body with the exception of the lungs. Whereas the right ventricle only has to pump blood into the lungs, which is, a low resistance, very high compliance structure. All right, and again, so this is kind of, again, a review to take you through all those steps I just mentioned. You have blood returning from the head and the upper extremity, which are, all those veins are coalescing into what we call the superior vena cava. And then from the trunk and the lower extremities, all of them are coalescing into the inferior vena cava. And they are both going to connect to our right atrium right here. So blood is going to, move directly into the right atrium. And separating again that right atrium from the right ventricle is going to be our tricuspid valve, or our right AV valve. When that valve is open, blood can move into the right ventricle, and then the right ventricle is going to contract, and it's going to move blood past what we call the pulmonary valve. The pulmonary valve is just what separates the right ventricle from the pulmonary circulation, or specifically the pulmonary trunk. And then the pulmonary trunk divides into the right and left pulmonary arteries, goes to the lungs, oxygenates. So that's why you see this returning from the lungs, which is going to be the left and right pulmonary veins, okay, which carry oxygenated blood to the left atrium. In the left atrium, again, we have our mitral valve, otherwise known as our left AV valve, into the left ventricle into the aorta. What separates the aorta from the left ventricle is going to be our aortic valve. The pulmonary valve and the aortic valve, okay, this is what separates the ventricles from the circulation. They're also known as semilunar valves. Okay, and lastly, I just wanted to mention quickly on that last slide that you'll take note that the blood flow is actually in series. So everything goes from the right side over to the left side, and they're all connected to one another. In other words, right atrium to right ventricle to the lungs, so left atrium, left ventricle into the systemic arteries, which just tells you that they're all in series. If anywhere along that series something is disrupted or blocked, that can cause backup of problems, since they're all interconnected. And in cardiology, we'll get more into how and why that is. Okay, but again, if your lungs are involved, your heart's involved, you can see things in patients like shortness of breath and distension of blood vessels because fluid is getting backed up because it can't get past a certain point. All right, and this is all again because everything is in series. Now, on the surface anatomy of the heart, you can also see sulci, and the sulci can actually help to actually indicate what the structures are internally as well. So here we have what we call the atrioventricular sulcus. The atrioventricular sulcus is right here in this region, also known as the coronary sulcus, and it's a groove that separates the atria, which you can see here. This bluish structure right here is the right atrium, and it separates that from the ventricle, which over here is this bulk of tissue right you see there is the right ventricle. So this little groove is between those two, and in fact, and I'll erase markings right now, is that in that groove, the sulcus there, you can actually see vessels running on the very surface of the heart there, and those are the coronary blood vessels. All right, both arteries and veins, and so these vessels, they run in those sulci, and they're typically on the surface of the heart, surrounded by adipose tissue or fatty tissue, which is the yellow tissue that you're seeing in the sulci there. Then there's also the interventricular sulcus. The interventricular sulcus is the sulcus that's between the two ventricles. It separates the right and the left, and you can see that on the surface of the heart. So over here, this is the interventricular sulcus. So that would be this portion right here. I'll draw the line there. So what happens is this whole region right here, again, is the right ventricle. This little segment over here, which is separated by the sulcus, is actually the left ventricle. So I can point out here that the apex of the heart is actually usually part of the left ventricle, not the right. So we only see a portion of the left ventricle here. The rest of it, again, is more posterior, which you can't see. But this interventricular sulcus separates it, and running on the surface of the heart through that sulcus is the blood vessels, all right, the coronaries, and they're surrounded by adipose tissue as well. Okay, so now I want to talk a little bit more about those valves. A couple things about the valves. First off, the valve separates the atria from the ventricles, and we refer to those as AV valves, or atrioventricular valves. The right side of the heart, it has a specific name called the tricuspid valve, and that's because it has 3 cusps. All right, those 3 cusps, when the valve is shut, separating the right atrium from the right ventricle, they come together at what we call the commissures. And then the left side of the heart, that AV valve, we refer to most commonly as the mitral valve. That only has 2 cusps. And these are mechanical valves, meaning that they open and shut simply due to pressure changes in the heart. So in other words, pressure changes between the right atrium and right ventricle dictate whether or not it's going to basically fall open or slam shut. Okay. There's no motor units or anything that are specifically causing them to close on their own. They only close in response to pressure changes. So otherwise it's essentially like a loose kind of, or it's just like a connective tissue that's kind of hanging out there, and when the pressure changes, it slams shut or opens. Then you also have the semilunar valves. The semilunar valves are the pulmonic valve and the aortic valve. And so those are the valves that separate the ventricles from the circulation. So the right ventricle from the pulmonary circulation and left ventricle from the systemic circulation. Now, over here we can see the right atrium, and here's the right ventricle. It's separated there. You can see the right AV valve, the tricuspid valve. And it's tethered to the inside of the right ventricle via these tendinous cords, also known as the chordae tendineae. And those chordae tendineae are tethered into the right ventricle in papillary muscles, which is written over here, all right? So these are papillary muscles. And these basically act like strings on a parachute, if you will. So in other words, when the right ventricle is relaxed, the tricuspid valve is open and loose. But then when the right ventricle fills up with blood, it's going to contract as it's generating pressure. I don't want to send that blood backwards into the right atrium. I always want blood to flow in one direction. I don't want it to go backwards. So what's going to happen is that generates a lot of pressure. That increase in pressure in the right ventricle is going to cause the cusps of the tricuspid valve to want to flop backwards into the right atrium. But instead of like flopping completely back into the right atrium, what happens is it stops and just closes at those commissures, or at the tips where all the cusps come together. It stops, and it stops because the chordae tendineae have them tethered, just like the parachute cords. And so that helps to keep them from blowing back into the right atrium when the pressure goes up. And then as the pressure goes up, that's going to cause these pulmonary valves, the semilunar valves. It's going to cause them, there's 3 cusps of the semilunar valve or the pulmonic valve, and that's just going to open up against the inside of the pulmonic trunk there and allow blood to move in. And its job is to prevent any blood from going from the pulmonary circulation back into the right ventricle. Because again, I want it to move in one direction, through the lungs to the left side of the heart, but not backwards. Once it gets to the left side of the heart, I have my mitral valve over here. And the mitral valve is similar to the tricuspid valve in its function. It lays inside the left ventricle when the left ventricle is relaxed, but when the left ventricle is contracting, and as it starts to generate more pressure, I don't want that blood going back into the left atrium. I want it to go into the aorta. So it's going to slam shut. The chordae tendineae are going to hold on and make sure that it doesn't billow back into the left atrium. And then that blood's going to move into the aorta after it opens up the aortic valve, which separates the aorta from the left ventricle. And that aortic valve's job is, when the left ventricle's pressure goes down, its job is to slam shut to prevent any blood coming from the aorta back into the left ventricle. So these are one-way valves that maintain blood flow in one direction, and that's their purpose. And so you can see that in the cartoons over here. If this is the blood entering the right atrium, as the valve opens, because the pressure in the right ventricle has decreased, blood will fill up the right ventricle, and as the pressure increases inside the right ventricle, then you'll be able to eject that blood into the pulmonary circulation. Okay, and you'll notice there's no backflow here of blood because we were preventing that by the shutting of those AV valves. Once you get the blood into the aorta or into the pulmonary arteries there, they don't go backwards because the semilunars or the pulmonic and aortic valves close and prevent any backflow. Now I'm talking about it in series here, but the reality is both the right and left side of the heart will be filling up with blood at the same time, contracting at the same time, and ejecting, so they're working simultaneously. So again, this is just kind of a quick review of the things that I just mentioned. You have one-way flow of blood through the heart, and that's the purpose of those one-way valves, okay? So those AV valves, right? The right AV has 3 cusps called the tricuspid, and then you have the mitral. Used to be called the bicuspid, just more commonly called the mitral valve. The chordae tendineae, all right, those are what actually connect those AV valves and tether them to the insides of the ventricles so that they don't blow back into the atria. And these semilunar valves, their job, again, is to control flow into the great arteries. In this case, that would be the pulmonary artery and into the aorta. And they'll open and close because of changes in pressure as well. So if the pressures go up in the right and left ventricle, that's going to cause them to open so that the blood can get ejected into those circulations. And then when they relax, we don't want the blood coming back in. Instead, they're going to slam shut and prevent blood from going backwards. All right. Okay, so now we're looking at the valves, but we're looking at it from the superior aspect of the heart. So it's like we're looking down on the heart. And actually, if you look at the cartoon over here, you can see the section. Basically, we remove the atria so that we can see all the valves. And if you take a look here, this would be the right AV valve or the tricuspid valve. So you can see the 3 cusps there. Over here, this is the mitral valve. The mitral valve is what separates the left atrium from the left ventricle, and it has those 2 cusps. Over here, you can see the aortic valve. And over here, you can see the pulmonic valve, which is labeled there in blue for you. Now, one feature I want to show you with this cartoon is that if we're looking here at the aortic valve, you'll notice it's got the 3 cusps of its semilunar valves there. And you'll notice on the wall, right over here and over here, it's got these 2 holes on either side that are just above the AV valve. Or excuse me, the aortic valve. So what happens is, as the left ventricle contracts, when it's generated enough pressure, the aortic valve will open, the blood will be ejected into the aorta. As the left ventricle starts to relax and the pressure goes down, blood's going to want to come back into the left ventricle, but it can't because the aortic valve is going to slam shut and prevent that. As it slams shut to prevent that, there's actually 2 holes on either side of the wall, 2 openings that you can see there, and those are the openings to the coronary arteries. These are the coronary arteries, as you can see. These are the arteries that feed the muscle, feed the tissue of the heart, so they're extremely important. This is how the heart gets its oxygen and nutrients. Okay. Now, in point of fact, despite the fact that it has blood going through all its chambers, it actually doesn't derive any real oxygen or nutrients from the blood that moves through its chambers. Instead, it has to rely on its coronary blood flow, its coronary arteries. Okay. So that's actually one of the first openings, or one of the first branches, if you will, off of the aorta. It's just above the aortic valve there. And so when the left ventricle is actually relaxing and the aortic valve shuts, that's when you're going to get good blood flow going through the coronary arteries there. And then, as you guys may already know, many of the myocardial infarctions and things like that are due to the fact that you have some sort of obstruction of flow in those coronary arteries that feed the muscles of the heart. That's just the heart's working all the time. It doesn't have a lot of reserve, so it requires, it requires a lot of oxygen frequently, right? And if there's any disruption in that, that can cause the muscle to weaken and even die, and that's what we refer to as myocardial infarction. So this is a review of the blood flow and the chambers and so. So something you can kind of quiz yourself with that I think works well. It gives you all the numbers there to it numbers them all so you can kind of go through it. because it's important to understand these flow and of course all the anatomy. So for example, you see point 1 over here. So this is blood that's basically entering the right atrium from the superior and inferior vena cava. All right, so it's the venous blood. And then the 2 right here, that's your tricuspid valve, which, if it's open, that'll allow blood to move into the right ventricle. From the right ventricle, when it contracts, it has to overcome enough pressure to open, of course, the pulmonic valve, which then allows the blood to move into the pulmonary trunk and into the left and right pulmonary arteries because it divides to go to each of the lungs. Once in the lungs, it gets oxygenated. That oxygenated blood is now coming back via the left and right pulmonary veins. There are actually 4 of them, so you can see 2 of them right here and 2 over here, which are a little bit posterior, so a little difficult to see. But they'll enter into the left atrium. So now we have oxygenated blood there. That blood will go into the left ventricle, and of course the valve separating that is the mitral valve. And then when the left ventricle contracts, it has to generate enough force to open up the aortic valve. And once the aortic valve is open, the oxygenated blood then moves into the aorta. And then when the left ventricle starts to relax, that aortic valve will slam shut to prevent any backflow. And so now you have blood moving in through the systemic circulation. Okay, so this is something that I know you guys are familiar with. I definitely talked about this in the earlier lectures, I think during the skeletal muscle lecture, when I talk about the structure of cardiac muscle. But I only did it in brief, and I'm just going to show you this again, is that cardiac muscle is striated, usually has a single nucleus, but instead of being long and cylindrical like the skeletal muscle, it's branched. So you can kind of see a little bit of that over here in this cartoon where they've highlighted. This would be one cardiac myocyte, and you see the nucleus in the center there, and you kind of see how its edges are jagged over here, all right? And so those are sort of branch points, and so what those will do is they'll actually interface with other cardiac myocytes, so maybe multiple different other ones. And so we refer to it as kind of branched, and you can kind of see that pattern there. And where they're connected there, we refer to those as intercalated discs, and that's the connection point between cardiac myocytes. If you look here at the microscopic view of it, you can actually see these intercalated discs all over. All right, there's these sort of dense bands that you're seeing there. And we refer to those as intercalated discs. Now again, that's where cardiac myocytes are being joined together. And if we take a look over here at this cartoon, you can see this structure here, which is supposed to represent that intercalated disc. And so there's proteins, what we call desmosomes. So these are like linker proteins that I mentioned from like the first lecture that help to keep the cardiac myocytes together, because all these muscles are going to essentially be pulling on each other. All right. And so they're interconnected like this. If I just interlock them, they have to be connected. So we need a lot of good, strong proteins like these linker proteins, like desmosomes, to help keep them together. And you see a high density of them at these intercalated discs. In addition, what you also have are these pores. And the pores are gap junctions. Now, I mentioned gap junctions in the nervous lecture because these are channels where essentially one cell can communicate directly with another cardiac myocyte via this gap junction. So there's basically an opening there, or a tunnel, where there's cytoplasmic connection from one cardiac myocyte to another. And if you recall from that nervous lecture, this is actually how I can transmit electrical signal very quickly. In smooth muscle, I talked about how it can, if they're linked by gap junctions, that can make them act as like a unitary contraction, where if one smooth muscle starts to set off an action potential, that can cause a whole host of them to contract essentially simultaneously. These gap junctions located in the intercalated disc are how one cardiac myocyte is going to communicate with another electrically, so that they will actually be coupled to contract effectively simultaneously. Okay, so in terms of metabolism of the cardiac muscle, it's almost exclusively aerobic respiration, and it requires obviously a lot of ATP because it is working your entire life, right? It's just constantly contracting and relaxing, contracting and relaxing. And if you think about it, if your average heart rate is about 75 beats per minute, all right, that's somewhere about contracting and relaxing in about 0.8 seconds or so. So it's working very hard all the time. And your heart rate when you exercise can go up to 120, 30, 40, or sometimes even higher. And that requires a lot of energy. So it does have a lot of myoglobin for some extra oxygen and some glycogens for extra nutrient storage. And again, it has huge mitochondria, which fill up like a quarter of the cell. Now, in terms of what it uses for fuel, interestingly, it actually prefers fatty acids under normal conditions. So normal, healthy working heart, 60% of its fuel is actually derived from fatty acids, which actually contain lots of energy. All right, so when they're catabolized, they'll be broken down into lots of ATP. Glucose is about 35%, and then it can utilize ketones and lactic acids and some amino acids, but to much less degree. What's interesting about this is something that we'll learn in cardiology, is that if the heart muscle is injured in any way, it can actually change what it utilizes. So instead of using just fatty acids, it might switch to more glucose utilization. And we actually use that information diagnostically to help us look at things in imaging. So a little foreshadowing, we'll come back to that. Okay, so now I want to talk about an action potential in a cardiac myocyte. This looks a little bit different from the action potential you guys learned about in a neuron, but the process is somewhat similar. There's just a few key differences. Now, with a cardiac myocyte, that makes up the bulk of the atria and ventricular muscle tissue, which is excitable tissue. They're connected via gap junctions, okay? Now, a couple things to notice on this action potential is, first off, the resting membrane potential of a cardiac myocyte is negative 90 millivolts. A neuron was about negative 70 millivolts. This is negative 90 millivolts, which is actually, if you guys recall, very close to the potassium equilibrium potential. Very close. So what that should suggest is that if my resting membrane potential, or the cardiac myocytes anyway, is negative 90 millivolts, and the potassium equilibrium potential is about negative 90 or negative 95, that means that what's permeable in the cardiac myocyte is primarily just potassium. There's very, very little sodium permeability here. If there were more sodium permeability, remember, we would reach a steady state between their two equilibrium potentials. The sodium's equilibrium potential would be something like plus 60 millivolts as it tries to get inside the cell. So they would come to some sort of steady state between them, and in a neuron that came out to be about negative 70 because potassium was more permeable than sodium, so it was closer to its equilibrium potential. In this case, we are even closer to potassium's equilibrium potential. So all that tells you is that potassium is definitely freely permeable, and there's relatively little, if any, sodium permeability at rest. So our resting membrane potential in the cardiac myocyte, again, is negative 90 millivolts. And we break up this action potential into phases. So this baseline here, where the cardiac myocyte is resting, is called phase 4. Phase 4 means that we have open potassium channels, as I just mentioned. So those channels are constitutively open, allows potassium to move freely, and it keeps the membrane potential at negative 90 millivolts. Now at this point, what's happened is there's been some sort of electrical excitation coming from what we call pacemaker cells, which keep the pace of the heart beating at a certain rhythm. And so when it gets the signal from that pacemaker cell, then you're going to see this sharp upward depolarization. Okay, what's happening there is that you are again activating essentially voltage-gated channels. All right, so we have what we call a rapid sodium influx through voltage-gated sodium channels. It's going to reach a peak somewhat above zero, so it's near 0 millivolts. It's going to be slightly positive right here. And then we're going to see this downward repolarization during phase 1. So phase 4, we were at rest. We got a signal from the pacemaker cell, which caused a rapid increase or rapid depolarization, which we call phase 0. And what that is it's voltage-gated sodium channels, and sodium is racing inside that cardiac myocyte, depolarizing it, making it more positive. So we get above 0, at which point those are closing or inactivating, which we went through. And then phase 1 starts. In phase 1, there's this transient potassium channels are going to open. So they transiently or temporarily open, and it allows potassium out of the cell. So as the cell is losing positive ions, it starts to become a little bit more negative, down to about 0, at which point we reach a plateau, which is from here to here, our phase 2. Now what happens is the reason we have a plateau is that we have potassium leaving the cell at the same rate which we are gaining calcium. So at this point, calcium, what we call specifically an L-type calcium channel, has opened. That L-type calcium channel, when it opens in the membrane of the cardiac myocyte, it's going to let calcium from the outside of the cell inside the cell, because there's a higher concentration of calcium outside of cardiac myocytes than inside. So what I have now is I have potassium, which is leaving the cardiac myocyte through its effluxing, and I have an influx of calcium. And since they're effectively in equal amounts at an equal rate, they kind of cancel each other out. I have positive ions coming in and positive ions leaving. They cancel each other out and create what we call a plateau effect. Okay, you can see that up here. Here's phase 2, and this is the ECF, ICF. Calcium is moving inside the cell as potassium is leaving outside, and then we reach that plateau. At which point the L-type calcium channels are going to close. They just inactivate, and the potassium channels remain open during phase 3. And since the potassium channels remain open longer, it continues to move out of the cell. So as it loses more and more positive ions, we start to repolarize during phase 3, and then we're back to phase 4. So again, in review, these cardiac myocytes all linked together by these gap junctions. Negative 90 millivolts is their resting membrane potential, all right, which is at phase 4. We reach threshold very quickly because of these gap junctions. So as long as the pacemaker fires, we then depolarize rapidly in phase 0, which is the voltage-gated sodium channels. They inactivate just above 0. Transient potassium channels open, which gives us a little dip in phase 1, a little repolarization. We reach a plateau in phase 2 as potassium channels are still moving out, but calcium channels are moving in now at the same time, and they offset each other in terms of charge difference. And so we end up with a plateau in phase 2, but then the calcium channels turn off. We go into phase 3, which is just the rest of our repolarization carried out by potassium channels. And so we go right back to our resting membrane potential at negative 90 millivolts. Okay, so here I want to talk about excitation-contraction coupling in cardiac myocytes. Now, this is actually very similar to what we saw in the skeletal muscle excitation-contraction coupling. But there are a few key differences. So what we're looking at here is a single cardiac myocyte in the cartoon. This is the sarcolemma, and if you recall, that's the plasma membrane of the muscle. So it's the plasma membrane of the cardiac myocyte. This is the extracellular fluid. That's just to help us get our bearings. Here's the cytoplasm. And here's the action potential, which is traveling along the membrane of our cardiac myocyte. And you'll notice that it has T-tubules, just like skeletal muscle. The T-tubules, again, these are invaginations in the surface of the cardiac myocyte so that it can quickly reach deeper structures like the myofibrils and the sarcoplasmic reticulum, so it can activate them more effectively. And so as the action potential travels along the membrane and hits those T-tubules, it's going to activate the receptors, and it's the same receptor that we saw in skeletal muscle, the dihydropyridine receptor. All right, that's the dihydropyridine receptor. And that dihydropyridine receptor, now here's where we get a little bit different from skeletal muscle. When this gets activated, this is voltage activated, so change in voltage causes a change in conformational state of the dihydropyridine receptor. In skeletal muscle, if you recall, that was mechanically linked to this receptor right here, which was the ryanodine receptor. So when there was a conformational change, it would mechanically alter the conformation of ryanodine, and that would open up the channel to allow calcium out. So these are the same two receptors, but their linkage is different. So as that action potential moves into the T-tubules and activates this dihydropyridine receptor on the surface there, that's actually going to open up a channel this time. It's not mechanically linked to ryanodine. So it opens up a channel and calcium comes in. So we have calcium that's in the extracellular space here. That's going to move inside the cell. That calcium is actually going to trigger the opening of the ryanodine channel. So when that's going to trigger that to open up, and it's going to cause the release of calcium. So this is what we call a calcium-induced calcium release. Basically, we've opened up a channel to allow calcium from the outside of the cell in, which then in turn activates ryanodine receptors. They open up and allow calcium from inside the sarcoplasmic reticulum to empty into the cytoplasm. And so now that calcium that leaves the sarcoplasmic reticulum will then interact with troponins located on the actin, move the tropomyosin. The actin is now exposed and the myosin can bind to it. In the presence of ATP, will go through its contraction cycle, just like we saw in skeletal muscle. So it's very, very similar, but the key difference here is we have calcium-induced calcium release. Now, some of the calcium that comes in from the dihydropyridine receptor, some of that will partake in the actual contraction of the myofibrils, but 90% of it will actually be utilized mostly just to open up the ryanodine channels to allow calcium to come in from the sarcoplasmic reticulum. Once the calcium has done its job and the contraction has taken place, obviously here we have this constitutively active calcium ATPase, which is pumping calcium back into the sarcoplasmic reticulum, similar to what we saw in skeletal muscle. And some of that calcium is also being pumped out through a sodium-calcium exchanger. The sodium-calcium exchanger means that calcium is being moved out of the cell as sodium is being moved into the cell. But we have to get the calcium out in order for the cardiac myocyte to relax. Now, the sodium coming in is going down its gradient, and that gradient, as you guys know, is kept by the sodium-potassium pump nearby. So that would make the sodium-calcium exchanger a secondary active transport mechanism. All right, so now I want to talk about the action potentials of the pacemaker cells. So these, again, these cells are specialized grouping of cells that are located in the atria of the heart. And so what they'll do is they'll actually spontaneously depolarize and fire action potentials. And the rate with which they spontaneously depolarize and fire action potential dictates the pace of your heart, or your heart rate. All right, so if it fires 75 times a minute, then your heart rate is 75 times a minute. So these are the guys that actually dictate that pace. And so they have special features on their membrane and special channels that allow them to actually depolarize spontaneously. So let's talk about some of those features here. So this is right here, this cartoon shows you what pacemaker cells, what their action potentials look like. And one thing you might notice right away is that there's no flat resting stage. So for example, I'll draw it up here. Oftentimes we see something flat. So this is the cardiac myocytes look like this, where phase 4 was flat and stayed at about negative 90 millivolts until it got depolarized and reached threshold at that point there. Here you'll notice that the phase 4 is actually right here. I'll draw it over here. You see phase 4, and it's the sloping upwards gradually. It's not flat, so it's not waiting for a signal. It's gradually depolarizing on its own. So it's going from negative 60 typically to about negative 40. Now negative 40 is actually the threshold. All right, so that's the threshold right there. Once it reaches threshold, then we fire an action potential. You notice this action potential is not as steep as the others, not as fast as the others. All right, and then of course it it goes a little bit above 0 here, and then it repolarizes back to negative 60, where it slowly depolarizes to threshold again. And so it repeats that pattern over and over and over again. It's because of what we call a pacemaker potential. The pacemaker potential is simply the slowly depolarizing baseline. It's also referred to as a pre-potential. It just means it's not steady and flat. It slowly depolarizes and then reaches threshold on its own. So let's go through what causes that. So here in phase 4, okay, you'll see that there is a slow sodium influx. So what happens is these are open channels to sodium that allow a little sodium to trickle in, and that's what we call it, sort of a slow sodium inflow. And then when it reaches about negative 50 millivolts, which is about halfway there, the threshold, then there's a slow calcium channel that opens and allows calcium to move in. So now we have calcium. So I'm going to put plus calcium over here since it's not labeled. Plus calcium start to move inside the cell and causes this gradual depolarization until we reach threshold. The negative 40 is a threshold for a voltage-gated calcium channels. Okay, it's a voltage-gated calcium channel. So I'm going to cross out sodium here. It's primarily a voltage-gated calcium channel. So they're going to open. And these are L-type calcium channels, and they're a little bit slower in their kinetics, but calcium is going to depolarize as it moves inside that cardiac myocyte to a little bit over 0, at which point up here it turns off. And then, of course, potassium, voltage-gated potassium channels have opened, and I'm going to get repolarization all the way back to my negative 60 starting point, at which point those voltage-gated potassium channels have closed off, sodium, and it starts to slowly move inside the cell again. Calcium starts to move in, reaches threshold, those L-type calcium channel starts to open, which are voltage gated, opens at negative 40, depolarizes, turns off, potassium turns on again, we repeat over and over again. So the main change here, really, there's a couple things to notice, is the spontaneously depolarizing baseline, which we call a pre-potential, or a pacemaker potential, and then that the upstroke, or the depolarization, is not carried out by voltage-gated sodium channels. It's carried out by voltage-gated calcium channels. So it's dependent on that calcium. We'll come back to that later, particularly in cardiology when we use certain medications that are calcium channel blockers. They can alter pacemaker activity because of it blocking that depolarization. Now lastly, let me erase some of this stuff here. I want to talk about 3 ways to alter the heart rate. So as we know, we can increase or decrease the heart rate, and that's due to our autonomic nervous system. And as I've discussed, the parasympathetic nervous system slows the heart rate down, and the sympathetic nervous system increases the heart rate. So this pace, of course, as it stands, if we're looking at this, let's say this is our normal here, all right? Every time it depolarizes, we're getting a contraction, then it goes to relaxing, and then it's contracting again. The rate with which it spontaneously depolarizes is dictating the heart rate, as I mentioned. Okay, and in a normal heart rate would be anywhere from 60 to 100 beats per minute. Now, let's say it got a strong sympathetic input. What's going to happen is you're going to now, let's say we're starting at negative 60, we have sympathetic activation. That's just going to increase the slope of the prepotential. It's going to increase the slope. So in other words, it can open up more, say, sodium channels and calcium channels to get it to depolarize faster. So we're going to reach threshold quicker. In addition, it could actually lower the threshold for those voltage-gated calcium channels and bring that threshold down a little bit so that the new threshold, maybe say negative 45 instead, and that allows not only a faster slope so we reach threshold faster, but now I bring the threshold down a little bit so I reach it quicker. And that will allow the heart to actually contract much faster. So for example, if I were to do that, I've lowered the threshold and increased the slope of my prepotential. I reach threshold faster, fire my action potential sooner, come back down, reach, come up faster, then reach it sooner, and so I can actually start to generate more and more beats over time. So this is how the sympathetic would stimulate these pacemaker cells. And of course, that would be activated via epinephrine or norepinephrine acting on beta-adrenergic receptors, primarily beta-1, right? And that would activate the G protein coupled receptor, increase in cyclic AMP, and the result of that interaction would be an increase in the prepotential slope and a reduction in the threshold, or making the threshold, lowering the threshold a little bit from negative 40 to say negative 45, something a little more negative. Now, if I take it and say, let's talk about the parasympathetic, parasympathetic would be the opposite, right? So one way it can do it is to increase the slope, so the amount of time it takes to reach threshold. So that's one way. Another way is to actually bring the starting point from negative 60 to negative 70. So it's further away from threshold, and actually take the threshold and even move it. So we can move the threshold up to say, let's say negative 30. Okay. start lower at -70 and decrease the slope. So it takes even longer for it to actually get there. And I finally fire an action potential. Comes back all the way down here, takes longer, and you can see that that would actually result in a much slower action potential. And so that would lower the heart rate. So an increase in parasympathetic input. This is via acetylcholine on muscarinic acetylcholine receptors in the heart, and its effect on the pacemakers would be, again, to take the resting, I shouldn't call it resting, but take the initial starting point from -60 to -70, make the threshold further away by from -40 to say -30, and then decrease the slope. And of course, that would be activating, G protein coupled receptors and so on. Okay, so now I want to talk to you about the order with which the current is directed through the heart. So for example, we talked just about the pacemaker cells on the last slide, and the pacemaker cells, they'll generate the action potential as we saw, and they'll dictate the pace of the heart rate. And when it's generating that action potential, all right, it's generating from this area over here at site number 1. That pacemaker site, those cells, that node is referred to as the SA node or the sinoatrial node. So the sinoatrial node, you'll see here, is actually located more in the right atrium, particularly where the superior vena cava and the right atrium come together. There's a little node right there called the SA node, and that SA node is generating the pace of the heart. And when it fires, remember, it's connected via gap junctions to cardiac myocytes all around it in the atria. So when it fires, it essentially spontaneously activates the atrial cardiac myocytes to fire their action potentials. Okay, and when they fire, of course, that's going to cause contraction of the atrial cardiac myocytes to contract in unison. And you'll see that's the red arrows. That's the movement of the conduction going through the atria, and it kind of moves like in a rippling effect, like a rock in a pond. It kind of ripples away from the node through the right atrium into the left atrium. And what it does is all those signals actually funnel through the atria, and they actually come together at another node. This node right here. Maybe we'll use a different color. See if purple works better. But this right here, and that's referred to as the AV node, or the atrioventricular node. This is another specialized group of tissue here that's between the atria and the ventricles. And all the signals from the atria are going to come together at the AV node. Now you'll notice they don't go directly into the ventricles. It doesn't just suddenly connect to the ventricles because there's separation between the atria and the ventricle. There's a connective tissue. All right, there's a connective tissue skeleton that actually separates the atria cardiac myocytes from the ventricular cardiac myocytes, so they're not linked via a gap junction. So when the sinoatrial node fires, it sends it only through the atrial muscle tissue, and that doesn't affect the ventricular muscle tissue. Instead, it funnels to the AV node. The AV node is how that electrical signal is going to get into the ventricles. So under normal circumstances, that connective tissue acts like an insulator and separates the atria from the ventricles electrically. That connective tissue also serves the purpose of actually helping the valves stay connected. All right, so it's where they're kind of tethered to as well. So again, that signal goes to the AV node, and then that initial segment there is called the bundle of His. All right, so it's called the bundle of His. Bundle of His. And then it divides. You can see it kind of divides to the left and right. Those are called bundle branches. All right, there's a left bundle branch and a right bundle branch because one bundle is going to the left ventricle, one's going to the right ventricle. So again, AV node, bundle of His, they branch into the left and right bundle branches. And something you need to notice here is that the bundle of His and the left and right bundle branches, that signal as it's moving into the ventricles is actually going through the interventricular septum. That's the muscular wall that separates the right and left ventricles from each other. So it's actually going down the septum first, which you're seeing here in 4. All right, so it's down this, and it's referred to as the AV bundle. So it's sending that electrical signal down towards the apex. So down towards the apex, where then the bundles split and go up the lateral walls. Okay, so AV bundle, bundle of His, that's going through the interventricular septum down towards the apex and then up the walls, up the lateral sides of the walls there. And then when it's going up the walls, it's turning into something called a Purkinje fiber. Right, so these are like very thin almost neuron-like type cells, and the bundle of His, bundle branches, and the Purkinje fibers will transmit that signal very quickly towards that apex and then up the sidewalls. Now, as I mentioned earlier, when you contract, you want to contract from the apex upwards. So that's why we want to send that signal down towards the apex as quickly as possible and start the contraction of the cardiac myocytes down there. So it travels to the apex, and that's where the contraction will start, at that apex. And then it'll slowly depolarize all the cardiac myocytes that are all linked via gap junctions to these Purkinje cells. It'll link, and it'll start a wave of depolarization of the cardiac myocytes up the lateral wall, so they'll start to contract from the apex up towards the base. In terms of the innervation, I talked about the sympathetic and the parasympathetic input to the heart, speeding it up or slowing it down, and it has an influence on the pacemaker cells for sure. So here's the SA node where those pacemaker cells are located. There's the AV node. Now the vega, the vagus nerve, cranial nerve 10, the right vagus nerve primarily innervates the SA node, and the left vagus nerve primarily innervates the AV node. And so it'll release acetylcholine, which the effect of that will be to slow the heart rate down. The vagus nerve primarily innervates, again, the SA and the AV node, as well as the atria, but there's very little parasympathetic input to the ventricle, to the ventricles themselves. The sympathetic input, now that's coming from that sympathetic chain that we discussed, okay, like part of the cervical and thoracic parts of the sympathetic chain. And that's going to form the cardiac plexus, and that's going to innervate equally the atria and the ventricles, as well as innervate the SA and the AV node. And the function of that will be to release norepinephrine to act on beta-1 receptors and to increase heart rate. So here, this is a little review of the conduction system that I just went through. Again, it's going to coordinate the heartbeat, right? So this is an internal pacemaker. Okay, that's our SA node, so that's the sinoatrial node. It's going to initiate them all from that prepotential baseline that just slowly depolarizes. That's located in the right atrium, and that spreads the signal out through the whole atrium via those gap junctions, which are connecting all the cardiac myocytes together. Okay, so now the SA node fires, signal goes through the atria, that all funnels to the AV node. The AV node is the gateway to the ventricles. So it's the gateway to the ventricles. Again, we don't send the signal directly to the ventricles from the atria. They're not linked via gap junctions. They have to go through this AV node. The AV node is actually going to cause a little bit of a pause, which is going to allow the atria to contract the blood and move it into the ventricles before we stimulate the ventricles. And again, that fibrous skeleton, that connective tissue that separates the atria from the ventricles, is going to act like an insulator that prevents any currents getting to the ventricles by any other route other than the AV node. And then we have the AV bundle, or the bundle of His. So this bundle, of course, then forks into right and left bundle branches, right to the right ventricle, left to the left ventricle, and it's passing through the interventricular septum. So it's going through that septum towards the apex. And then, of course, that's going to communicate with the Purkinje fibers, which are these nerve-like processes that spread throughout the ventricular myocardium. So the Purkinjes are kind of intertwined through all the ventricular myocardium at the apex and up the sidewalls there, and that's going to very quickly send action potentials to the cardiac myocytes, and it's going to send that signal via gap junctions again with the cardiac myocytes in the ventricles, and that'll start to initiate contraction at the apex, and then as the lateral walls become activated, they'll start to contract as well. So the sequence goes from apex towards the base. Okay, so this cartoon I actually just got from Wikipedia here, and what I like about it is it's showing you some of the things we just talked about. Like here's the SA node. That's the SA node that's firing, so that's the pacemaker, and that's dictating the heart rate. And when that fires, all the cardiac myocytes linked to it in the atria are going to depolarize, and you can see that wave of depolarization. And where it goes to is right here, and that is the AV node. And there's a little bit of a pause there, and then the AV node, when that fires, that's going to send the signal down the interventricular septum towards the apex and then up the walls there via the Purkinje fibers. So remember, connected to the AV node is the bundle of His, Then there's a left and right bundle branch, which reach to the apex, and then you have the Purkinje fibers from there, which are intertwined with all the cardiac myocytes of the ventricular walls. And so you see that the contraction occurs at the apex and moves towards the base, which the base is up here. Okay. What you're seeing at the bottom below that, though, is actually an EKG or an ECG, which is how we record electrical activity in the heart and how it coincides. And you see this little blip right here, which we refer to as a P wave, actually coincides with the firing of the SA node and depolarization of the atria. So the P wave represents atrial depolarization. And then you have the QRS, which we call this structure right here. And the QRS has a greater amplitude. As you can see, it's much sharper in appearance. And that coincides with ventricular depolarization. And then finally, this last wave right here is called a T wave, and that just represents ventricular repolarization. And this is something we can record on the surface of the person's body via those electrodes, and this is the kind of pattern that we would see. And the only thing you may not see here is atrial repolarization, and that gets lost or obscured by the QRS complex there. Okay, so now this is the electrocardiogram, all right, as I kind of just mentioned on the previous slide, and we're actually going to go into a lot more detail on the electrocardiogram, otherwise known as an ECG or an EKG. We have a separate class for that, and we get into it in cardiology, where we'll get, a lot of opportunity to actually use that to make diagnoses. Some of you may be somewhat familiar with it already, and this is something where we put leads on the chest and limbs of the patient in order to record the electrical activity of the heart. And we can make certain diagnoses from that. So for example, somebody with chest pain, and we take an EKG, we could actually detect something like a myocardial infarction. It could even tell us so much as to where that's occurring in the heart. So we can get a lot of information. We can also figure out the rhythm of the heart from that, whether it's a normal sinus rhythm, we call it, or something like atrial fibrillation and so on. So it can be a very good, very strong diagnostic tool, and it's used quite frequently. So we'll get to know it more in detail later. But for now, we need to at least understand what the waves represent. So here's just like what we call a rhythm strip, where we can see multiple complexes over time. So this, along the x-axis here, is time. And so I can see, one complex here is another complex here, and another one, and so on. And they're spaced apart just so much. And over here it's saying about 0.8 seconds apart. And that's, the next one will be the same. It looks very regular. And we'll learn how to take a heart rate, but what this represents is every time you see one of these complexes represents, the heart beating. And so that's the heart rate. How often does it occur over time? If it occurred once every second, well, you'd have, a heart rate of 60 beats per minute, which, is at the bottom end of your normal heart rate, or the lower end. So it's 60 to 100 is your normal range. Now, if I take a look at one of these complexes down here, you can see it's broken up to the waves that I mentioned on the previous slide. The P, the QRS, and the T wave. Now, the P wave represents atrial depolarization, QRS, ventricular depolarization, and the T wave, ventricular repolarization. And the atrial repolarization, as I mentioned previously, is actually lost or obscured by that QRS complex, which is why we don't see it there. You'll also notice that the timing of when the P wave starts and ends, and the beginning of the QRS, and then finally with the T wave, they have what we call intervals. So we have something called the PR interval and the QT intervals, and there's a specific timing of this. It could be too long or too short, and that can be pathological. And the QT intervals, the time it takes from the start of the QRS to the end of the T wave, which is the entire ventricular depolarization and repolarization. So that entire duration. Again, there's a normal range for that. That's what we're going to learn about later on. And that can either be too long or too short. All right? And so these are things that we're going to kind of come back to. But just as a quick point here, let's say we're looking at the P wave, atrial depolarization. And then there's a little bit of a pause here. There's a little bit of a flat line, which you see right there. We refer to that as the PQ segment or the PR segment, which is right here. And so this is the pause at the AV node. So just after, at this point here, remember, all that depolarization through the atria is going to reach the AV node, and then there's a little bit of a pause. So that's when it reaches the AV node right there. Then there's a pause. You see that pause, and it has a certain length of time to it. All right, that little segment there. And then the QRS is the depolarization of the ventricles. So that's where the AV would pause there, and then it would subsequently depolarize. So as an example, if it takes too long, if that segment is too long, we may have some problem with the AV node. It's not acting quickly enough. And oftentimes we refer to that as something like a heart block, which we'll come back to. If it's too short, we might be exciting the ventricles too quickly. And so these are things we'll be looking into later on, and how to sort of interpret what that is. So this slide is just to demonstrate how the ECG, what you're seeing on the ECG, those waveforms, how it coincides with what's happening within the heart. So here's the P wave, and you see the depolarization, again, wave of depolarization occurring in the atria. All right, once it's fully depolarized, you can see that there. And then you'll notice that the QRS, again, there's a little bit of a pause at the AV node, and then the AV node fires, and then you start to see the start of the QRS as it's going down the interventricular septum towards the apex. And then here you see the complete QRS of the entire ventricles are depolarized. And then it starts to repolarize from the apex upwards, and you can see that in the form of the T wave. Now the cardiac cycle is what I want to talk about next. So this is, in the cardiac cycle, this is everything from the filling up of the ventricles with blood to their contracting and ejecting that blood and then refilling up with blood. So from the time it's relaxed to contracting to relaxed again, that's one cardiac cycle. And this, again, occurs if you have a heart rate of 60 beats per minute, it's occurring once every second. The average person is typically about 75. So this, you're looking at a cardiac cycle occurring in less than a second. So the cycle of events from relaxation to contraction actually has specific names. So anytime the heart is contracting, we refer to that as systole. So the heart contracts. Now with systole, when I say heart contracts, what I mean is actually you have atrial systole. So the atria contract separately from the ventricles. So when the atria contract, that's atrial systole. When the ventricles contract, that's ventricular systole. Now, actually, oftentimes when I just talk about systole without specifying atria versus ventricle, I'm really referring to the ventricles, because that's the one that actually, under normal circumstances, matters more. But if I'm referring to the atrial systole, I'll refer to it as atrial systole. Otherwise, when I say systole, I'm referring to the ventricles. Relaxation. When the heart muscle relaxes, we refer to that as diastole. So systole and diastole. So diastole is the relaxation portion of it. All right, and so that's what I've labeled down here, although systole and diastole refer to contraction, relaxation of either type of chamber, atria or ventricle. It's typically referring to ventricles, so keep that in mind. Okay, so I'm actually going to draw out for you the cardiac cycle. And I apologize in advance for my drawing, so I want to make sure that you guys are all on the same page here. So this is the heart that I've drawn, and I'm going to be drawing everything from the perspective of the right side of the heart. And the reason I'm doing that is for clarity's sake, because in the cardiac cycle, everything I'm about to talk about from the perspective of the right side of the heart is exactly the same as what's happening on the left side of the heart at the same time. So if there are any differences, I will mention them, but overall you can assume that whatever I'm talking about is exactly the same as the left side. So that's why I'm only focusing on the right side right now to make it a little bit easier and to make sure we understand some of the stuff that I've drawn in here. This right here is the superior vena cava. This right here is the inferior vena cava. They are draining via the red arrows, the blood, into the right atrium. And this is the right ventricle. And it's separated by this structure here, which is the AV valve. In this case, specifically, that would be the tricuspid. And then the right ventricle is connected to the pulmonary trunk and separated by that semilunar valve known as the pulmonic valve. And of course, it bifurcates into the pulmonary trunk bifurcates into the left and right pulmonary arteries. So in this phase, what I want to show you is, to start, ventricular diastole. So ventricular diastole, recall that diastole is relaxation. So this is the ventricles. Both ventricles are relaxed right now, meaning that they're not contracting at this time. So what's happening now is that while they're relaxed, they're going to fill up with blood. So when they relax, the tricuspid valve and the mitral valve would open up and blood would start to move inside the ventricles. Now the amount of blood that actually fills up those ventricles, or the volume, is known as the end diastolic volume, or EDV. And that's basically the total amount of blood that fills up the ventricle during diastole. We call it end diastolic volume because at the end of diastole, at the end of the period with which it's relaxed, how much blood does it have? And that is equal to 130 mils on average. Now 130 mils in each ventricle, not total, in each ventricle is 130 mils. Now diastole is actually broken up into phases. So the first phase, the beginning phase, is rapid filling. So in rapid filling, what that means is that the tricuspid valve has just opened up and the blood that is accumulated in the right atrium is now pouring quickly into the right ventricle, for example. So that's rapid filling. And then it goes into what we call a slower phase, a slower filling phase, which is kind of trickling in more. And that's referred to as diastasis. And diastasis is the middle portion of diastole, where it's gradually filling. And then we have the last part of diastole, which is atrial systole. So this is when the atria is actually going to contract and eject any remainder blood into the right ventricle or into the left ventricle. So it's going to contract and eject that last percentage. And that last bit of blood makes up about 20% of the total volume coming from the atria. So 80% or more is coming from the rapid filling and the diastasis, and that last 20% is being ejected via atrial contraction. So if we take a look at it back here, what that means is that, again, we have the tricuspid is open, so there's communication between the right atrium and the right ventricle, or the left atrium and the left ventricle. Blood is now filling initially passively into the right ventricle and into the left ventricle. The volume reached in each of the ventricles is going to be 130 mils on average at the very end of diastole. So that's after the atria has contracted and ejected that last bit of blood. The total that's going to be in those ventricles is about 130 mils each. And the pressure in the right ventricle and the left ventricle is very low at this point. It's low pressure. So this is actually something I should mention now, is that blood flows down its pressure. It goes from high pressure to low pressure. All right, so it has to have low pressure. So in other words, it's relaxed, it's not contracting, the muscle's not as tense, and so the chamber, the inside of that chamber there, the ventricle, has a lower pressure. So now here's step 2. And in step 2, we are in something called early ventricular systole. So early ventricular systole. So what that means is now the ventricles, this is the beginning part of when the ventricles are going to start to contract. Okay? So when they contract, you'll notice something that I changed in the drawing over here is the AV valve starts to close. What happens is, as the ventricular muscle starts to contract, all right, that cardiac muscle starts to contract, and I drew that with these little arrows here pointing inward, basically to indicate that the chamber is going to get a little bit smaller as the muscle around the wall starts to contract and squeeze inward. And so as it starts to do that, it's going to generate pressure. All right, and this early contraction, we actually call this isovolumetric contraction. We call it isovolumetric because what's happening is as the pressure is beginning to increase in the ventricles from the contracting muscle, the volume inside the ventricles hasn't changed yet. We haven't ejected any blood yet. Okay, but what does happen is as we start to contract, the AV valve closes very quickly, and so it slams shut. So that tricuspid valve or that mitral valve closes and quickly closes it off, so that way blood doesn't go directly back into the right atrium or into the left atrium. So that has to close, and in fact when that closes, that produces what we call an S1, which is actually the first heart sound we hear. So you might recall, maybe if you've ever listened to the heartbeat, it's got really two beats to it. It's got what we call an S1 and S2, or otherwise called a lub-dub, 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 lub-dub. Those two sounds are actually coming from closure of the valves. So that first heart sound you hear is actually the simultaneous closing of both AV valves. So S1 is both AV valves shutting. And so that's shutting to prevent blood from going backwards, and that happens because pressure is starting to increase in the ventricle as it's contracting. And so it slams shut. All right, so it's slammed shut. So the volume right now hasn't changed. So the volume inside my ventricle, since it's isovolumetric, is still my end diastolic volume, which is 130 mils. That hasn't changed. All right, but now that we're starting to contract, that means the ventricles have started to depolarize. So if we're talking about the ECG, that coincides with the QRS wave. And as I mentioned already, the sound that you're going to hear is the S1 sound, which is the AV valves closing. That gives you that sound. And now, at this point, pressure in the ventricles is starting to rise. And as it's rising, you'll note that initially it's going to shut the AV valve. And just to quickly point out here that the pulmonic valve and the aortic valve would still be shut as well. So we're not ejecting anything either. So we're generating an increasing pressure in the ventricles, but blood can either move backwards or forwards at this point because there's not enough pressure to open up the pulmonic or aortic valves at this time. So the volume doesn't, so that blood doesn't go anywhere, so the volume stays the same as pressure is increasing. We would call that isovolumetric contractions. Okay, so step 3 is going to be late ventricular systole. So in late ventricular systole, so now this is, we talked about the early start of contraction, and now this is later into the contraction as more and more pressure is being generated by those ventricles. So you can see here, we're generating more and more inward pressure on those chambers. And we've increased the pressure enough. You'll notice what happens here is this. We still have a closed tricuspid valve. That's not going to open regardless of the amount of pressure we generate there under normal circumstances. That stays shut, so that way no blood is going backwards into the atria. However, once the pressure is significantly high enough, the pulmonary valve and the aortic valve will open. In other words, we have to overcome a certain pressure in order for the semilunar valves to open. Now in pulmonic circulation, the pressure we have to overcome is the pressure of the pulmonary circulation, which tends to be on the order of like 15 to 20 millimeters of mercury. And this is the pressure that needs to be generated by the right ventricle in order to open up that semilunar valve, because that's the amount of pressure that's keeping them shut. So it has to match that. So in the pulmonary trunk, for example, here, that would be about, let's say, 15 millimeters of mercury of back pressure keeping the pulmonic valve shut. So that means that the right ventricle has to generate at least 15, a little over 15 actually, in order to have enough pressure to overcome that back pressure. So it has to generate greater than 15 millimeters of mercury pressure in that chamber so that blood moves out of the right ventricle and into the pulmonary circulation. And that's the pressure that's required. The left ventricle actually has a much higher pressure that it has to reach. In fact, the left ventricle, and this is where the differences are occurring that I was mentioning before, that actually has to generate enough pressure upwards of about 80 to about 90 millimeters of mercury in order to open up the aortic valve, because the back pressure coming from the aorta to keep the aortic valve closed is about 80 millimeters of mercury. So it has to match and then exceed that in order to have enough pressure to push blood into the aorta and open up that valve. So that's what late ventricular systole is. It's reaching enough pressure. And when it does, we have what we call ventricular ejection. We've generated enough force, enough pressure inside that chamber to create a gradient so that we have higher pressure in the chamber of the ventricle, so it will flow into the circulation. So now again, on the right side of the heart, that means we have to generate about 15 to 20 millimeters of mercury. And in fact, we'll generate much more pressure than that. It doesn't just barely exceed the minimum, it actually goes above. So the pressure in the right ventricle will actually reach a maximum of about 25 millimeters of mercury. So we're well above the threshold of about 15 or 20. The pressure in the left ventricle is going to max out at about 120 millimeters of mercury, which is well above 80 millimeters of mercury, which is the minimum amount to open up that valve. So that's the maximum pressure, is about 120. That's a big difference in pressure from the left side and the right side of the heart, because the left side has to overcome more pressure because it's ejecting it to the systemic circulation. The right side of the heart is only ejecting it into the pulmonary circulation, so it doesn't require as much pressure. Now another key here is the amount of blood that actually is ejected. What is the volume? It doesn't eject, each ventricle does not eject 130 mils. It doesn't eject the whole volume. In fact, the volume that's actually ejected, all right, the volume is 70 mLs is ejected. 70 is ejected out of the 130, so 70 mils is ejected out of the total 130 mLs. This 70 mLs is actually referred to as stroke volume. So that's the volume of blood that's actually ejected from the ventricle when it contracts. That's the volume that enters into the pulmonary or the aortic circulation. In both cases, the volume is the same. It's 70 milliliters on average out of that whole 130 that it started with. Which means if we take a look at that ratio that I just drew, 70 over 30, that's about 70 over 30 is equal to, that's equal to about 0.54. If I multiply that by 100, that's 54%. So 54% of the blood was ejected into the circulation. What I just calculated for you there, 54% was just ejected, is actually referred to as the ejection fraction, otherwise known as the EF, ejection fraction. And a normal ejection fraction for people is somewhere between 50 and 65% of their volume in the ventricle. So if it started with 130 and they ejected 70, they have an ejection fraction of 54%, and that's within the normal range. All right. Now, we normally don't want to eject the entire volume because we want to have some reserve. This is the ejection that's occurring at rest. If we start to actually exercise or something like that, our heart's going to contract harder, and it's going to eject, larger volumes. And in fact, it could eject up to about 90% of the total volume. But this gives us at least some room to eject larger volumes if necessary. And so now in the last phase here, phase 4, what we're reaching is what we call early ventricular diastole. Early ventricular diastole. And so now we're relaxing again. This is the early phase of relaxation. So actually what I was speaking about in the very first part of the cycle was ventricular diastole, and I spoke to it as a sort of a whole. But now when the ventricle is actually done contracting, it's going to start to relax. Okay, and as it starts to relax, we go into what we call isovolumetric relaxation. Isovolumetric relaxation. So again, this is the same term, isovolumetric. It means the volume is not changing as it's relaxing. So recall that if it started with 130 mils, it ejected 70 mils. All right. So it's left with 60 mils, and that's 60 mils over here. All right, that volume is not going to change. What's actually going to occur first is as the pressure starts to release, as it starts to come down. That's why I drew the arrows facing outward. It's starting to relax. The chamber's getting a little bit larger as it relaxes, so the pressure is dropping. As that pressure drops, I'm reversing the gradient. So what's going to end up happening is now the pulmonary, the blood that I just injected into the pulmonary circulation, that's going to want to come backwards and go back into the right ventricle, because now the pressure is higher inside the pulmonary circulation than it is in the right ventricle. And the same goes for the aorta and into the left ventricle. It wants to come back now since the pressure is dropping. And what happens is now those semilunar valves are going to close. They're going to close in response to that and prevent that backflow. So there's no blood that's coming back from the circulation. But the pressure is still too high to allow the AV valves to open. So the mitral and tricuspid are not open yet because the pressure is still too great. So it's relaxing, but now both the valves are still closed. So no volume is changing, so we call it isovolumetric relaxation. So this is where pressure decreases without a change in volume until the pressure drops below the pressure in the atria, at which point those AV valves will open and the cycle will repeat. But until it gets to that point, all right, it's still isovolumetric. And so what we have here is this volume, and the volume is what we refer to as the end systolic volume. It's the volume left in the ventricles after it's ejected the stroke volume, or in this case, the 70 mils. So what's left is, as I mentioned, 60 mLs, because we started with 130 and we lost 70. And so there's 60 that's left in both the ventricles. And we're repolarizing as we relax. So on the ECG, that coincides with the T wave. That coincides with the T wave, and the shutting of the semilunar valves coincides with the heart sound of S2. So the sound you're hearing is S2, and that's the closing of the semilunars. And then finally here, what you're seeing is just a repeat. This is actually the same cartoon that I drew in the very first, the first drawing. And so when the pressure finally drops enough, we're back now in ventricular diastole, where the heart is now, the pressure has dropped enough in the ventricle that the tricuspid and the mitral valve will open, and blood will then start to fill up the ventricles again, going through the same phases that I talked about: early diastole, diastasis, and then atrial contraction, and we'll repeat the whole cycle again. And everything I just discussed in all those cartoons, if your heart rate's about 75 beats per minute, then this all took place in about 0.8 seconds or so. So this, again, the amount of time it takes for this to happen varies with the heart rate. So the higher the heart rate, the faster this occurs, right? The lower the heart rate, and so on. Okay, so I just finished going over a visual description of the cardiac cycle. And in that, we talked about how the ventricular pressures change during diastole versus systole, how the volumes change during diastole and systole. And so what I want to show you here graphically is how the pressures change. So we need to be able to interpret this graphically, and I'm going to draw this from the left ventricular perspective. So the pressure graph I'm going to draw is the pressure changes that occur in the left ventricle during systole and diastole. Understand that the right ventricle would be exactly the same curve, only with lower pressures. And as you can see on the y-axis there, I have pressure in millimeters of mercury from 0 all the way to 120, and then it's over time. Okay, so for example, in the left ventricle, when the left ventricle is in diastole, the pressure in the chamber is very low. So I'm going to draw something low, pretty close to 0, and fairly flat. It's relaxed. At this point, it's filling up with blood. Okay, but then right at the end of ventricular diastole, there's a little blip in pressure. And that little increase in pressure there that you see actually coincides with the atrial systole. Because if you recall, at the very end of ventricular diastole, when it's about to start contracting, the atria actually contracts and ejects that last bit of blood into its chamber and that contractile force where it ejects another 20% of blood into the ventricle causes a little increase in pressure. All right, but it's a transient increase in pressure. And then what happens immediately after that is the ventricle is then going to proceed into its systole or systolic phase. And when that happens, the pressure goes up sharply, up to about 120, comes back down again, and then baseline. And so this is approximately what the curve would look like for left ventricular pressure. Again, the same curve for right ventricular pressure, only the peak would be much lower. So a couple things to take note of is that during this time from here to the start all the way to before the pressure really rises, this is all part of diastole. Okay, so that's all part of the ventricular diastole. So it's filling up, and again, that little blip over here that you're seeing in pressure just represents atrial systole, so it's ejecting that last bit of blood into it. And then at this point, I'm going to circle this point right here with the green. That's when the pressure starts to rise. That's when we enter into ventricular systole. So as the pressure begins to rise, that point that I just circled, that's when the AV node, or more specifically for the left side, would be the mitral valve, when the mitral valve closes. Again, that's to prevent backflow. So as pressure starts to increase inside the left ventricular chamber, the AV valve, or the mitral valve, is actually going to slam shut to prevent any backwards flow. Because again, we always want it to move in one direction towards the systemic circulation, and not backwards into the atria. So the AV valve closes to prevent that as the pressure is going to increase. Now the pressure is going to rise, and if you recall, it's going to rise until it reaches a high enough pressure where it can overcome the pressure of the closed aortic valve, which is approximately 80 millimeters of mercury. So I'll draw a little dotted line to that point there. So that point there represents how much pressure has to be generated before it can open the aortic valve. So I'll draw this here. This point right here represents opening of the aortic valve. And in fact, from the time the AV valve closes down here, all the way till we reach this point right here, where the aortic valve is going to open now, and we can eject blood, that increase in pressure where there's no volume change is the isovolumetric contraction. So the isovolumetric contraction, that's where we're increasing pressure in the chamber, but both the valves at this point are closed until the pressure gets high enough where the aortic valve will open. On the right side, that would of course be the pulmonic valve would open. And so 80 millimeters of mercury, we reach that, now the valve opens, and so we're ejecting blood once we reach pressures above that, and oftentimes in normal, healthy individuals, that pressure will increase to approximately about 120 millimeters of mercury. And I'll continue to eject blood that whole way up until we reach about 120. Then you'll notice that we go on the downward portion of the curve here. And that's when the ventricle starts to relax now. So it's the sort of early portion of diastole, if you will, when it starts to relax. And in fact, as it starts to relax, to prevent backflow of blood that just was ejected into the aorta, to prevent that from coming back into the left ventricle, the aortic valve has to close. So right around this point right here, the aortic valve closes. And so that prevents backflow back into the left ventricle. And the ventricle will continue to lower its pressure more and more, until at which point we will reach approximately here. And at this point, the pressure is low enough now, lower than what's in the atrium, and so the AV valve will flop open. So the mitral valve will just open up because the pressure in the left ventricle is now lower than what's in the left atrium. And so that flops open, and now blood can then empty into and fill up the left ventricle from the atria. And so this in total shows you how the pressure changes. And if we keep in mind the cardiac cycle and what's happening with the valves, you can actually mark off, as I have already for you, when the valves are opening or closing and when isovolumetric contraction, and that reminds me, isovolumetric relaxation, which of course occurs when the aortic valve closes from when the mitral valve or the AV valve opens. So this would be isovolumetric relaxation. And again, that isovolumetric relaxation means the volume is not changing as the pressure is decreasing inside that chamber, but both of the valves are still closed. Once the pressure drops low enough, the mitral valve will open and the atria will be able to empty its contents into the left ventricle, and we have a repeat of the same process we just talked about, which again will be in diastole with a little blip at the end and then another rise and so on. So this process will continue to repeat once every second, or once every 0.8 seconds, depending on the person's heart rate. Okay, so I'm continuing to look at pressure changes from the left side of the heart here, and I'm just going to quickly redraw the left ventricular pressure graph. But what I want to add now is I want to actually add another pressure tracing, which I'm going to draw here in red. Okay. And so to make this a little bit clearer, okay, so the tracing that I just made in red actually represents the aortic pressure. Down here is the left ventricular pressure graph. So how do the pressures change in the aorta? So what we're seeing here is that the aortic pressure steadily declines. Okay, you can see that it steadily declines. But at this point, this junction right here, which is right about the 80 millimeters of mercury point, right? That's when the AV valve, or excuse me, the aortic valve opens. And so blood is now being ejected into the aorta. This is where the left ventricle is now communicating directly with the aorta. It's open to it. So any pressure that is generated by the left ventricle at this point will then be transmitted directly into the aorta, which is why you see the graphs essentially become one now, and they follow the same trajectory here upwards, because they're effectively communicating openly now because the valve is open. So the pressure in the aorta will go all the way up to 120 millimeters of mercury, matching the pressure that's being generated by the left ventricle, since the valve is now open and communicating with, then the two chambers are now communicating with each other. And as the pressure starts to decrease as well in the left ventricle, initially the aortic valve will match that closely as well, except at this point right here, because what's happening right there is now the aortic valve closes. The aortic valve is closing. And so what you see is a temporary rise, a little transient rise in the pressure right there, as some of the blood that's trying to make its way back to the left ventricle gets blocked by the closure of that aortic valve, and you get a little surge in pressure in the aorta there. And then you see again it has this gradual decline in pressure, and this occurs because of the elasticity of the aorta. When you're injecting a large volume into it, it stretches and distends, accommodating all that extra volume, and then it starts to slowly squeeze that blood, and since it's elastic, come back to its original position, and as it does so, it squeezes that blood forward into circulation, and so the pressure gradually declines because of that until we hit again the aortic valve opening because of the contraction of the ventricle again. Then it repeats over and over again. So in the aorta, we see this pulsatile pressure where it goes from about 120 to 80 before the next ventricular contraction comes along, then it goes up to 120 again, then gradually back down to 80. And in fact, you may recognize these numbers already, but if you don't, I'll point it out right now, is that 120 over 80 is our average, or our healthy blood pressure, is approximately 120 over 80. So that's 120 millimeters of mercury over 80 millimeters of mercury. This is exactly the same thing we're checking when we check an arterial blood pressure in somebody's arm with a blood pressure cuff. So when you're reading 120, and we say that's the systolic, and this is the diastolic pressure, what they're referring to is the systolic and diastolic pressures that are in the artery. And so at 120, that's the peak pressure, because right now it's open to the left ventricle, which is ejecting a large volume of blood into it until the pressure rises. But then as the left ventricle closes, and its pressure, as the left ventricle is closed off, from the valve, and its pressure is declining, the pressure inside the aorta gradually declines as well, but not to the same extent. Obviously, it's not dropping to zero. It gradually declines to about the level of about 80 millimeters of mercury before the next contraction occurs. So it does not drop to zero, but 80. So we have what we call a pulse pressure. The difference between the systolic and the diastolic is about 40 millimeters of mercury. And that's our average for our blood pressure. Okay, so that's the left ventricle and the aortic pressure graphs. The last one I'm going to cover right now, again, if I'm drawing the left ventricle, its pressure graph, so on. All right, so now what I want to draw for you is what the left atrial pressure looks like. And so left atrial pressure, remember in diastole, ventricular diastole, it's emptying into it passively, and so its pressure is going to be approximately the same. So I'm drawing it essentially on top. That's the red graph right here. So it's going to be left atrial pressure. And when it contracts, it of course contracts to the same amount, or the pressure that's increased in the left atrium is to the same extent that we saw the increase in the left ventricle. And so they follow essentially identically so far. But then, as you guys recall, at this point here, the left ventricle is going to increase its pressure as it starts to contract and go into systole. And so therefore, this is actually the point again to remind you that the AV valve is closing to prevent backflow into the atria. So what's going to happen here is we're going to see what we call a little blip, and then it gradually increases and then comes down and repeats. And so we're seeing something that during the systolic phase, while the left ventricle is contracting and in systole, we're in diastole for the atria. It's essentially just passively filling up, as in this case, the pulmonary veins would be emptying that oxygenated blood into that left atrium. And on the right side of the heart, it would be the superior and inferior vena cava emptying the systemic venous blood into the right atrium. And so when the AV valve closes, that closure causes a little bit of a snap or a bulge back into the left atrium, which is why we're seeing this little initial blip. And then this steady incline upwards is actually the pressure increasing as it's filling up passively with volume coming from, again, the lungs or the systemic circulation. And then over here you see the pressure drops, and that drops right there because the left ventricular pressure is now less than what's in the atria. And so here is where the AV valve opens again. And so when you see the pressure drop from the left atrium there, it's because now it's emptying its contents into the left ventricle, wherein now it's open to the left ventricle, and so their pressures become the same during diastole once again, and then the whole process repeats itself. Okay, so this is actually what we call a Wiggers diagram, and you'll notice actually right away that the very top portion of the Wiggers graphs here is the pressure, which we actually just talked about, pressure over time, and how the changes in the left ventricle, left atria, and aortic, how they all change over time during the cardiac cycle. So that's what I reviewed already with you guys. So you can take a look at that again for a review. What I like about the Wiggers diagram, though, it also includes ventricular volume. Okay, so for example, you'll notice the volume start from 60 and go to 120 mLs. So I mentioned about 130 mLs in my previous examples, so that's an approximation. All right, so here, going into diastole, excuse me. Remember, that's the flat portion of this curve right here, which I'll circle up here. So as we go into diastole, you'll notice we're starting with 60 mLs already in the ventricle. That's the end systolic volume. So that's after it's ejected its blood, what's left in the ventricle is about 60 mLs. And so since we're heading into ventricular diastole, the atria is going to empty its contents there. So it's passively emptying its contents. And then it's going to contract and eject that last bit. And you'll notice that comes to a peak of about 120 mLs in this example. So it gained about 60 mLs from the atria in total. And then you'll notice we kind of flatline at this point right here because the AV valve is closed and we're in isovolumetric contraction, which on the pressure graph is this portion. And then when the aortic valve actually opens and we eject, the volume in the left ventricle is going to decrease. It's going to decrease, and it actually drops back to about 60 mLs. So we ejected 60 mLs during that contraction. So the stroke volume would be 60 mLs. And we're now down to our what we call our end systolic volume again, which is what we started with, which is since we had 120, we ejected 60, we have 60 left over. So that's our end systolic volume. And then the process repeats itself. The atria open up, it passively empties into it, contracts that final volume, and then we go through the whole process again. I also like this because it shows you how the ECG coincides with each portion of this graph. So for example, the P wave. The P wave is atrial depolarization. Well, when the atria are depolarized, they're going to contract. So that actually coincides with this little blip there, that contraction of the atria. The QRS, when the QRS depolarize, what follows shortly after that is contraction of the ventricles. And if you look up, you'll see that that coincides with contraction of the ventricle. The pressure increases in the left ventricle. And then as the left ventricle is relaxing, you'll notice the T wave. It coincides with the T wave during repolarization, when everything is resetting itself and it's relaxing. So it shows you how the ECG aligns with all that. And then lastly, the heart sounds, which are going to be useful, right? So during the QRS phase, when the left ventricle is starting to contract, I told you guys that the AV valve closes, and that first closing sound is called an S1, and that's audible, something we can hear with our stethoscope. And then when the aortic valve closes, we hear the S2. And of course, that would be aortic and pulmonic together at that point would be the S2. So we see how everything kind of lines up. So this graph is excellent for looking at the cardiac cycle and understanding it in terms of volume, ECG, heart sounds, and pressures. So I highly recommend spending some time getting to know this particular graph. It will definitely help you moving forward.